Sa bawat kwento ng pag-unlad ay may mga institusyong nagsusulong nito. Institusyong nagbabahagi at tumutugon sa mga hamon ng panahon.
Picard supports S&T Human Resource Development. Nagsusulong ito ng mga programa na naglalayong mapaunlad at mapayaman ang kakayahan ng mga researchers natin sa bansa at mga empleyadong katulad ko for better delivery of S&T products and services.
Sinusuportahan namin ang ating mga dalubhasa na kaisa natin upang umasenso ang ating bansa.
Tumutulong kami na maging makabuluhan ang mga teknolohiya upang magbigay ng karagdagang kabuhayan sa ating mga mamamayan.
Nagbibigay kami ng mga bagong kaalaman at teknolohiya na gagabay sa pagkalinga ng ating kalikasan at magpapaunlad sa kabuhayan ng mga magsasaka at mangingisda.
Isinusulong namin ang paggamit ng agham at teknolohiya bilang pagsagot sa mga hamon ng lipunan.
Kasama ninyo ang DOST Picard sa pagpapaunlad ng sektor ng agrikultura, aquatica at pangisdaan at likas na yaman. Sa gabay ng agham at teknolohiya, kami ay nakatuon sa pagpapataas ng produksyon ng pagkain, sa pagpapabuti ng kalidad ng pamumuhay lalo sa kanayunan, sa pag-aalaga at pagpapatatag ng ating kalikasan, at sa pagpapalakas ng kapasidad at pamamalakad sa pananaliksik at pag-unlad. Ang DOST Picard ay nangunguna sa pagbibigay ng kakayanan sa pagsasaka, aquatica at pangisdaan, at panglikas na yaman upang makipagsabayan ng sektor sa pandaigdigang pamantayan.
Narito po ang DOST Picard upang tulungan ang ating mga magsasaka, mangingisda at ang nasa sektor ng likas na yaman sa pamamagitan ng agham at teknolohiya. Isinasagawa ng DOST Picard ang aming mga programa sa pamamagitan ng pakikipag-ugnayan sa iba't ibang ahensya ng gobyerno, state universities and colleges at mga international partners. Ito ang DOST Picard. Kami ang DOST Picard.
Good morning, everyone. Welcome to the NSTW Technoflora 2022, hosted by the Philippine Council for Agriculture, Aquatic, and Natural Resources Research and Development of the Department of Science and Technology, or DOST Picard. From the Crops Research Division, I am Danica Sembrano.
And from the Inland Aquatic Resources Research Division, I am Dana Lilogronio, and we are your hosts for this morning.
Sa ating pagdiriwang ng National Science and Technology Week, 3 research initiatives ang ating ibibida kaugnay ng temang Ensuring Food Security or AANR Technologies, Busog-Lusog na Nasyon sa Agri-Aqua Innovation.
Ang Agriculture, Aquatic, and Natural Resources or AANR ay mahalagang parte sa buhay ng bawat Pilipino. Kaya nga excited na akong malaman kung papaano makakatulong ang agham at teknolohiya sa AANR, lalo na para sa ating mga entrepreneurs, researchers, magsasaka at mangingisda sa buong bansa.
That's right, Miss Dana. Pero bago natin formal na simulan ng ating Technoflora, nais po naming batiin ang ating mga manonood ngayon ng live sa DOST Picard Facebook page at sa World Trade Center, Metro Manila. Right now, we are joined by about 84 live viewers, kasama pa ang ating mga media partners mula sa iba't ibang bahagi ng bansa.
Marami rin sa ating mga kasama ngayon ay mula sa iba't ibang state universities and colleges, local universities and colleges, private universities, as well as secondary schools. Thank you so much for being here with us today. And we encourage everyone to tag friends in the comment section so that they too will be updated with these research initiatives that will be shared with us this morning.
At syempre, inaanyayahan din namin ang lahat na sumali sa ating live open forum mamaya kung saan sasagutin ng ating resource speakers ang inyong mga katanungan.
So now, to formally open our Technofora, let us hear the welcome message of DOST Picard's Executive Director, Dr. Reynaldo V. Iborra.
Our esteemed speakers from Mariano Marcos State University, University of the Philippines Visayas, and Mindanao State University, Tawi-Tawi, students, guests, and participants watching through our official Facebook page, a pleasant morning and warm welcome. Every year, we look at the National Science Technology Week as an opportunity to showcase DOST Picard's outputs and outcomes of its industry strategic S&T programs or ISPs through exhibits and fora. Science and technology has become part of our everyday life, and this fact is highlighted in this year's theme of ensuring food security AANR technologies, Busog-Lusog na Nasyon sa Agri-Aqua innovation. The technology fora is only a teaser of how science and technology impact on various areas addressed by traditional disciplines, including the management of agriculture, aquaculture, and natural resources. This activity will highlight the results generated from the different S&T activities and provide us information on the advances in the AANR. Topics reflect the DOST Picard R&D initiatives in agriculture, aquaculture, and natural resources technologies for dissemination. This is also an opportune time for us to share our views, bright ideas, and knowledge in the emerging challenges on science and technology to be adopted by our Agri-Aqua sector and other stakeholders to boost the AANR industries in the country. Today, the topic for the Technofora comprises of 3 completed projects. Topic 1 covers the development of low glycemic index rice through induced mutation and marker-assisted selection under the leadership of Ms. May Rose Mauriat Abad of MMSU. The project was conducted to develop low glycemic index rice through induced mutation and marker-assisted selection, which is acceptable to the consumer preference. Mutation was induced to NSEC RC160, a popular peel rice bred irrigated rice cultivar with good eating qualities. 5 mutant populations were generated using physical and chemical mutagens and a combination of both agents. Topic 2 covers the organic bioactive aquafeed additives for efficient and sustainable aquaculture, led by Dr. Rex Ferdinand Tripalgar of UP Visayas. In this presentation, organic bioactive feed additives developed by the Institute of Aquaculture UP Visayas through DOST Picard funding will be discussed, including the practical application to improve growth, disease resistance, and food conversion efficiency of cultured aquatic animals. Natural and organic feed additives isolated from seaweeds and aquatic microbes will be presented as growth enhancers, metabolism boosters, and stress and disease resistance activators in cultured aquatic animals. Lastly, topic 3 covers the Seaweed Research Center in BARM under the leadership of Dr. Siti Saida Halon of MSU Tawi-Tawi. The CERDEC consists of a seaweed culture lab, a molecular genetics, and seaweed post-harvest processing laboratory. The center combines laboratory, hatchery, and farming technologies with morphological, genetic, and physicochemical analysis of farm and wild Capopicus species and Eucoma denticulatum varieties across the Sulu archipelago. The center has collected more than 1,000 wild and farmed eucumatoid samples and prepared a seaweed GIS distribution map in the Sulu archipelago. Through this fora, we are expecting to identify other researchable areas and additional technology interventions for future AANR research. It is further hoped that aside from the attainment of the forum objectives, this will be an avenue for a fruitful exchange of ideas and discussions and potential R&D collaborations. Allow me then to take opportunity to recognize the efforts and give my sincere thanks to the organizers, the Inland Aquatic Resources Research Division, Marine Resources Research Division, and Crops Research Division staff who have been working hard even with multiple tasks and responsibilities. This event would not have been possible without them. We are hoping that this activity will further develop the agriculture, inland aquatic, and marine commodities, improve our technologies, and further ignite our zeal for R&D towards ensuring food security. Thank you, and I wish you all a very productive day.
Thank you so much, Dr. Iborra, for that warm welcome and for introducing the topics that will be discussed this morning. Sa puntong ito, mukhang handa na ang ating mga viewers na mas makilala pa ang ating first speaker.
All right, our first speaker earned her master's degree in molecular biology and biotechnology in the University of the Philippines, Los Baños, and her bachelor's degree in biology in Mariano Marcos State University. She has been involved in various research initiatives on rice, especially during her time as science research specialist in the Philippine Rice Research Institute.
She is currently a professor at the Mariano Marcos University and the project leader of the development of low glycemic index rice through induced mutation and marker-assisted selection. Here to present her topic, Low GI Rice, the Present and Future of a Healthier Rice, live from Ilocos, let us all welcome Ms. May Rose Maurat Abad. Good morning, Ma'am May.
Good morning
Morning,
po.
ma'am.
Good morning sa inyong lahat.
Hi, Ma'am.
Morning, ma'am. Ma'am, tanong lang po namin sa inyo para po sa kaalaman ng ating mga viewers. Ano po ba yung ibig sabihin ng low glycemic index?
Ang glycemic index ay sukatan ng mga pagkain na may carbohydrates base sa kanilang probabilidad na ito ay maging sanhi ng pagtaas ng ating blood sugar level. Kaya pag sinabi natin na low glycemic index, ibig sabihin ang mga pagkain na ito ay mas healthy para sa atin, lalong-lalo na sa mga taong may diabetes.
Thank you, Ma'am May, for answering our question. Mukhang interesting itong ibabahagi mo sa amin, lalo na sa potential na benefits ng low glycemic index rice sa kalusugan ng bawat isa. Kaya wag na nating patagalin pa. You may take the stage, Ma'am May.
Thank you po.
A pleasant day to all the participants of this year's National Science and Technology Week Technoforum. It's an honor to present the result of our project funded by the USTP card titled Development of Low Glycemic Index Rice through Induced Mutation and Marker-Assisted Selection. This is led by Ms. Teodora Imananghaya of PhilRice Central Office in Muñoz, Nueva Ecija, with yours truly as the co-lead, Ms. Ria Yate, Mr. Riomel Kim Albano, and Ms. Janet Polipol of PhilRice Batak as researchers under the project. I am Mayros Emawirat Abad of Mariano Marcos State University, here to present to you Low GI Rice, the Present and the Future of Healthier Rice. So just to give you a rundown of our presentation, I will be giving a brief introduction about the project and about low glycemic index rice, the objectives of the project, the methodology, the results, and a summary of the findings that we had, our future plans or our ways forward. So to start with, rice is still the most important food crop in the world, specifically in the developing countries such as the Philippines. This is evident with the increasing per capita consumption of roughly 110 million Filipinos, from 92 kilograms in the 1990s to 111 kilograms in year 2019, or 2009 rather. 119 kilograms in 2019, and last year, 2021, a total of 133 kilograms of rice is consumed by each Filipino citizen annually. So with this data, indeed, rice is life. This increase in rice consumption can be linked to rapid urbanization and market strategies such as only rice in fast food chains and restaurants. With this, increase in cases of diabetes is of primary concern. So diabetes is the fifth cause of death among Filipinos in 2020 and the years prior. During the pandemic, diabetes ranked fourth as the leading cause of death in the Philippines. This can be attributed to the change of lifestyle and eating habits we had during the pandemic. And the most common food that we had then were rice plus other carbohydrate-packed foods such as instant noodles, which is a staple from the ayudas we received. Hence, it's no surprise that diabetes moved a step higher in the ranking. So in this presentation, we will be talking about glycemic index. But first, what is glycemic index? Glycemic index, or GI for short, is the value assigned to carbohydrate-rich foods according to its relative effect on the blood glucose level after consuming that food, or what we call the postprandial blood sugar. In reference, the GI value of glucose is 100. In rice, the GI value of freshly cooked rice ranges from 64 to 93. As a rule of thumb, if the rice is soft when cooked, it has high GI. But if it is hard, it is healthier, it has lower GI value. According to the Sydney University Glycemic Index Research Service in Sydney, Australia, low glycemic index food has a GI value of 1 to 55. Brown rice has a GI value of 50 to 55, which is considered low. White rice, on the other hand, or polished rice, or the most common rice that we had or is available in the market, ranges from 64 to 93. The range is wide because some polished rice are soft, some are hard or firm when cooked. Again, the rule of thumb, the harder or firmer the rice, the healthier it is. Glutinous rice, on the other hand, has a GI value of 90, which is high. Yet still, as Filipinos, cook this sticky rice into kakanin, adding sugar, molasses, and syrup, which adds more to its GI value. In a study by Bravad Harini, the consumption of white rice is associated with the increased incidence of diabetes in Asia. This is the reason why brown rice is advocated for people suffering from diabetes. Studies also show that brown rice can help in preventing diabetes, lowers stress in lactating women, boosts heart health and nervous system. It is rich in antioxidants, helps prevent Alzheimer's disease, improve digestive health, controls cholesterol level, and it has antidepressant properties. However, brown rice is more costly than ordinary polished rice, or the white rice as we know it. And additionally, it is not widely available. Plus, brown rice cannot be stored longer than 6 months. This is due to the oil present in the bran, and the bran layer is the one that is removed during polishing. So this is also one of the reasons why brown rice is costly, since they are packaged in small proportions only compared to the packaging in polished rice, which are sold per sack. So hence, this project was crafted. The project on the development of low glycemic index rice is led by PhilRice Batak in collaboration with PhilRice Central Office and MMSU. The project is supported by the USTP card under the leadership of Dr. Reynaldo Ebora and funded under the USTP card GIA. It's a 3-year project which started last 2020 and will terminate this year. The project aims to develop low glycemic index rice through induced mutation and marker-assisted selection. Specifically, the project aims to determine the effects of the different mutagenic agents on the genotype and phenotype of NSIC-RC160, identify mutant lines with increased aminose content and decreased glycemic index through marker-assisted selection, and identify the perception of consumers on low glycemic index rice. So for the methodology, mutation was induced into NSIC-RC160. RC160 variety is a PhilRice bred variety released in 2007. Old but gold as it is still one of the most preferred variety of consumers because of its superior eating qualities. As market is consumer driven, RC160 is also a variety preferred by our farmers. 2 types of mutation was used to generate the mutant population: physical and chemical mutagens. Gamma irradiation was used for the physical mutagen with 2 doses, 250 gray and 300 gray, and sodium aside at 1 millimolar concentration was used for the chemical mutagenesis. To maximize the induction of mutation, double mutagenesis was also done. So this is done by combining both the physical and chemical mutagens. Hence, 5 mutant populations were generated. For the molecular screening, STR markers were initially used to check the diversity of the population. The STR markers used are listed in the Lusog's article, stating that this list of markers are enough to determine the genetic diversity within rice population. For the selection, trait-based SNPs were used for different grain qualities such as amylose content. So these data were analyzed using Power Marker version 3.25. Alongside with the line advancement and molecular screening, the mutant population were also characterized agromorphologically. After the advanced generation or advancement of the generation, grain quality evaluation was also conducted. We also did survey to know the consumer's perception on low glycemic index rice. And the survey was conducted in 10 HUCs based on the Cities and Municipalities Competitiveness Index or CMCI in 2019. So in our survey, we surveyed respondents in 4 strata. So we have college students, members of the academy, policymakers, and healthcare professionals. So we have 2 per strata, so a total of 48 respondents per HUC. So we chose this group of individuals because of their technical understanding of the technology and to low glycemic index, and their perception could possibly reach and influence a larger and wider group of consumers later on. So basically, NSIC-RC160 was mutated to produce 5 mutant populations. And from this mutant population, selection was made using genotypic and phenotypic approaches to identify improved lines based on its morphology, yielding abilities, but most importantly, on its grain qualities. So for the results of the genotyping, A slight polymorphism was observed between the mutant lines. So results revealed formation of 3 major clusters with a similarity of 40 to 93.33%. Cluster 1 include a single genotype, M248, and cluster 2 with 2 genotypes, M333 and M357. While the rest of the population, along with the NSIC-RC160 wild type, clustered together. So this shows that diversity, or there is, the lines were really diverse, and that these different types of mutations applied affected the diversity of the NSIC-RC160 mutants. So line M248 is from the bean MO3 population. The bean MO3 population was applied with chemical mutagen, whereas the 2 lines that clustered in cluster 2 were from the bean MO4 population. This population were applied with a combination of the chemical and the physical mutagens. So to further identify the lines that makes the population different from the rest of the mutant lines, agromorphological characterization was done at M4 generation. This figure shows the NSIC-RC160 wild type side by side with a representative line from bean MO4 and bean MO3 population. Based on the data gathered, the plant height of the mutants under bean MO3 and MO4 are considered dwarf, whereas NSIC-RC160 is intermediate based on the standard evaluation system for rice and the plant variety protection guide. For the grain length, the mutants were classified as long grains, while the NSIC-RC160 wild type has medium grains. So this shows high phenotypic variability between the mutant population compared to the wild type in terms of the grains, the length of the grains, and the plant height. However, for other traits such as tiller number and panicle length, low phenotypic variation was observed. Okay, so these are example panicles from the mutant lines showing the effects of the mutation and the morphology of NSIC-RC160. As mentioned in the previous slide, panicle length has low phenotypic variability between and among the lines. So the length of the panicle did not significantly change even when these lines are subjected to different types of mutagen. So trait-based genotyping using SNPs were done, and the SNPs, these traits were based on grain quality related markers. It was done at M5 generation, and the result of this genotyping was used to identify the lines to be advanced and will be subjected to grain quality analysis. So based on the result, 20 lines were selected, and here are some of the promising low GI lines. Based on the results of the grain quality analysis, 4 mutant lines had improved rice or head rice percentage or head rice recovery compared to the wild type. So the wild type is classified as grade 1, but the mutant lines are classified as premium quality. So what is head rice recovery? Head rice recovery is very important during the post-harvest process, as higher head rice recovery means lesser loss during post-harvest processing. But most importantly, another notable result of the grain quality analysis is the increase in amylose content from low in the wild type to intermediate and high in the mutant lines. As mentioned a while ago, NSIC-RC160 was chosen because it is highly preferred by consumers due to its superior eating qualities. And this is because it has low amylose content. Low amylose content means softer texture of rice when cooked. Hence, the high preference of consumers. We all love soft textured rice. However, low amylose content in rice also translates to high GI values. Thus, the increase in amylose content in the mutant lines suggests a lowering of the GI values of these or in these mutant lines. So let's move to the result of the survey and consumers' perception. So again, a group of college students, members of the academe, healthcare professionals, and policymakers from the 10 HUCs in the Philippines based on CMCI 2019. These cities include Makati, Manila, Muntinlupa, Pasig, Quezon City, Bacolod, Iloilo, Davao City, and Cagayan de Oro City. Results showed that the respondents were unfamiliar with rice varieties. Most of the respondents associate rice brands to rice quality, and the most common to them were rice labels and brands such as Sinandoming, Dinorado, Jasmine, and others. These are brand labels usually available at the supermarkets and groceries in packages. In addition, respondents were only familiar with the term white rice as a variety. Hence, survey found heterogeneity of rice preference, showing evident diversification on white rice and specialty rice. So, when the respondents are asked on their preferred rice quality, qualities like soft and aromatic are most preferred by these respondents, while 19% of them go for healthier choices, and the rest settle for anything that is available in the market or in their locality. When the respondents are asked as to their daily rice consumption, 62% of the respondents consumes 2 to 3 cups of rice daily. 3 cups was observed to be highest in the groups of healthcare workers and members of the academe. And surprisingly, the least in the four groups to consume 3 cups of rice are the group of students. Additionally, data revealed varying results across different cities. Cities like Bacolod, CDO, Makati, Muntinlupa, Pasig, and Quezon City consumes an average of 2 cups of rice daily, while those in Iloilo consumes only an average of 1 cup of rice daily. For the awareness on low glycemic index rice or low GI in general, results showed a low level of awareness to glycemic index. Majority of the respondents are not aware of the concept of GI and its effects on diabetes prevention and management. So 16% of these, 60% are from the policymakers and 10% for the members of the academe. However, level of GI awareness was found to be the highest in the groups of healthcare workers. And this is not surprising since they are in the medical field and the GI term or glycemic index term is considered as a medical jargon. Thus, information on glycemic index must be fed on schools and government institutions to spread awareness. For the summary of findings, 20 mutant lines were selected based on molecular and morphological data, and these lines are already in the pipeline for crop improvement. The combined irradiation and chemical mutagens have significantly affected the genotype, morphology, and quality of rice. There is an available market for low GI rice. A survey conducted finds consumers from highly urbanized cities prioritize health over price in their decision and purchasing power. Moreover, the results opens an entry point for possible intervention on the consumer level in these urban areas. the potential effect or information should be considered in campaigning for GI rice or low GI rice. Information dissemination and campaigning on the awareness and consumption of low glycemic index rice must be strengthened, especially in schools and government institutions. So at present, we have a problem on diabetes and its management. And so we have this project as a way of helping Filipinos have better and healthier rice choices. For the future of low GI rice, some promising lines are already in the pipeline, and we hope that these lines will be released as a variety soon. While the determination of GI values of food is expensive and uses human subject, other ways to determine the GI values includes an enzyme assay test. And we now correlate the result later on onto the GI values using a standard sample. So our group is in the process of procuring materials for the enzyme assay test to determine the quantitative values of GI of these mutant rice lines. So later on, we will determine if these lines really have low GI. Also, alongside with this seed multiplication of the 20 initially selected lines is being conducted, and once they are seed produced or seed multiplied, this will be used for sensory evaluation later on to test if these mutant lines with higher amylose content is acceptable to the consumers. Because remember, when we have higher amylose content, the texture of the rice will be hard or firm. So let's check if the ones that we will be producing will be acceptable to the consumers. Also, another round of SNP genotyping is also seen. This time is for different biotic and abiotic stresses. So we also want to know if these lines have improved tolerance to different biotic and abiotic stresses. And with all the data that will be gathered from these above activities, this will be used for potential plant varietal protection application of the mutant lines and inter-yield trials, hoping that this will become a released variety in the future. And once available, crafting of policy recommendation for adoption is seen as well. So before I formally end this presentation, I would like to take this opportunity to thank DOST card for funding our research, the CRD team most especially, and special mention to Dr. Edna Anit and Sir Lito for all the help when we are starting with this project. Your contribution to this project will be forever be remembered, and your contributions will always be forever valued. So thank you so much. Thank you for listening.
Thank you, Ma'am May, for that very interesting and informative presentation. Siguradong maraming natutunan ng ating live audiences on your presentation on the development of low glycemic index rice. Ikaw, Miss Dana, any takeaways mula sa presentation ni Ma'am May?
Alam mo, I learned na inversely correlated pala ang GI sa amylose content. And high amylose content means firm rice texture. Pero sa project nila, Ma'am May, they were able to develop acceptable rice texture to consumers. Ikaw ba, Miss Danica?
Ako naman, I'm just very excited na successfully mailabas at maipromote itong low GI rice dahil bukod sa maganda yung eating quality nito, ay healthy pa ito. So ito ay magandang balita talaga para sa ating mga Pilipino, lalo na't malaking parte ng ating buhay ang kanin. And to our audiences, if you have any questions addressed to Ms. May, you may type in those questions sa ating comment section para masagot iyan mamaya ng ating expert during our open forum. Just remember to type your name, affiliation, or location.
We are also now live at the World Trade Center, Metro Manila, and we would like to greet everyone listening there. Now, we will turn the time over to our second speaker. He is an expert in aquaculture and he specialized in nutritional immunology during his PhD in Kagoshima University, Japan. He is currently a professor at the Institute of Aquaculture, College of Fisheries and Ocean Sciences in the University of the Philippines, Visayas.
To talk about the organic bioactive aquafeed additives for efficient and sustainable aquaculture, let us all welcome Dr. Rex Ferdinand M. Trifalgar, live from Yagao, Iloilo. Good morning, Sir Rex.
Magandang umaga
Hi,
po sa
sir.
lahat, sa ating lahat na nakikinig at participant sa event na ito. Magandang umaga po. Good morning.
Good morning po. Sir Rex, ano-ano po itong mga aquafeed additives na na-develop ng UPV?
Marami po tayong na-develop na mga additives with the project of the DOST collaboration with UPV and the public sectors and industries. These are all intended to increase the efficiency and to lessen the use of fish meal and to improve productivity sa ating mga cultured aquatic animals po.
Thank you, Sir Rex. Mas po kaming na-excite, sir, dahil ibabahagi mo din sa amin ngayon ang mga advantages ng aquafeed additives. So, go ahead, sir.
Thank you. Good afternoon, everyone. Today, I would like to present to this group my talk entitled Organic Bioactive Aquafeed Additives for Efficient Good afternoon, everyone. Today, I would like to present to this group my talk entitled Organic Bioactive Aquafeed Additives for Efficient. Good afternoon, everyone. Today, I would like to present to this group my talk entitled Organic Bioactive Aquafeed Additives for Efficient and Sustainable Aquaculture. So, I am Rex Ferdinand Trafalgar, a faculty of the Institute of Aquaculture, College of Fisheries and Ocean Sciences, University of Philippines, Visayas. So as we know, the country is currently experiencing a fish shortage. This is basically due to the low cuts coming from the municipal fisheries or the low production from our municipal fisheries as well as from the commercial capture fisheries. So currently, aquaculture is expanding and contributes to about 41% of the total fish being produced in the country. And the statistics tells us that about 1.3 to 1.4 million tons of fish, we are short of 1.3 to 1.4 million tons of food fish per year because our total per capita consumption exceeds our total production. And this is also compounded as an effect of the declining catch from the capture in municipal fisheries. And it is expected that aquaculture should provide the needed fish in order to fill the gap and to fill the 1.3 to 1.4 million tons of fish shortage. However, aquaculture is highly dependent on feeds, and the profitability and economic sustainability of aquaculture is basically linked or dependent to feed cost that is related to the operation cost of the of this economic activity. Before, aquaculture is highly dependent on fish meal as the major ingredient of all formulated diets for aquaculture. However, it has been regarded as an inefficient way to utilize fish resources since there is a need to capture fish and use that one to feed another fish. So currently, the trend is to develop plant-based material to be converted into a high-protein feed ingredient to be utilized as a component of fish diets. Basically, fish requires a higher protein than terrestrial animals as part of their diet. And most terrestrial plants would not satisfy the requirement of aquatic animals. But the trend now is to convert these carbohydrate, cellulosic based plant materials from the terrestrial ecosystem to be used, then convert this one into a high protein based material so that it could be utilized for aquatic animals. So basically, this talk would be focused more on the ways to minimize fish meal utilization in aquatic animals in the use of feed additives and fish meal alternatives to limit the use of fish meal in the formulation of feeds for aquatic animals. So the beauty of bioconversion is that with biotechnology, it is possible to convert plant-based biomass that are rich in carbohydrates and cellulose into a material that could be high in protein that would equal soybean meal or other feed legumes, and use that one to be a part of the component of fish diets. So one of the solutions is biotechnology. Fermentation could be used to increase the protein content of algae as well as carbohydrate-rich material, including copra meal and sweet potato meal, to increase the protein content of the biomass so that it could be used to replace or to be used as ingredient to replace soybean and fish meal in aquatic animal diets. So the first dietary ingredient that I will be discussing would be the pro-NK or protein enriched protein enhanced kamote. So this product was developed by Tarlac Agricultural University in collaboration with Plentex Philippines as well as UPV. So we have fermented a sweet potato biomass that very rich in carbohydrates into a material that equal the protein content of soybean meal. The sweet potato now would contain 18 to 40% protein with a lipid content of 3%, a fiber content of 6 to 7%, and a carbohydrate content of 45 to 63%. And we found out that heavy metals are not present, or if present, it is on the acceptable level on this fermented material. And the digestibility of the developed ingredient was found 71% for tilapia, 98% highly digestible for milkfish, 98%, and it is highly digestible with shrimp, about 86%. It is better than soybean meal in terms of digestibility. So the sweet potato that normally would contain 3% protein, we have converted that one to about 40% by fermentation. And it has better digestibility than soybean meal for the major aquatic animals that are being tested on this study. So the amino acid profile was found to be complete, though there have been the material is short in terms of methionine, lysine, and arginine, but these essential amino acids are still present in this material. The fatty acid found on this material were almost medium chain and short chain fatty acids that are highly important or has a growth promoting bioactivity for aquatic animals, because these fatty acids would not be processed in the liver but could be channeled directly to every cells of the fish or aquatic animals because they are medium chain and short chain fatty acids. So we tested incorporating this diet to the diet of tilapia, ranging from 0 to 50% incorporation in the diet. Then we formulated diet containing this pro-NK, and we fed to milkfish, tilapia, and shrimp. Then we found out the results suggest that weight gain is improved in diet containing 50% inclusion of this feed ingredient. The FCR is also improved in those that receive the higher inclusion levels. So in milkfish, higher inclusion level promoted growth, at the same time better feed conversion as compared to the control group. For tilapia, similar trend could be observed. Higher weight gain were found on those that received the higher inclusion level. The feed conversion data also shows in tilapia that it is better at the higher inclusion levels. In the result of Pineos Baname, growth promotion is significantly exhibited in the highest inclusion level as compared to the control group. And feed conversion is significantly enhanced also in the highest inclusion level of 50%. Because Pineos Baname utilizes carbohydrate material better as compared to milkfish and tilapia. And other than carbohydrate, this ingredient contains short-chain fatty acids that also promotes muscular growth and better nutrient assimilation in this shrimp. So the carcass composition of the 3 aquatic animals were found to be similar. So there is no decline in protein, lipid, and carbohydrates content on the carcass or on the flesh of these aquatic animals, indicating it is not affecting the nutrient composition of the flesh of these aquatic animals being tested. But there is a significant increase or improvement in the ratio of omega-3, omega-6 fatty acids in these aquatic animals, indicating that the inclusion level of fermented sweet potato improved the deposition of omega-3 fatty acids in the flesh and tissue of these aquatic animals. Because an increase in omega-6 is an indicator that the animals are not of good quality, and consumers don't like flesh of animals with significant amount of omega-6. But these dietary treatments containing this fermented sweet potato promoted the deposition of omega-3 to be significantly enhanced as compared to omega-6 in the flesh of these cultured tilapia, shrimp, and milkfish. There is a decline in omega-3, omega-6 ratio, and there is an increase in the content of omega-3 fatty acid in the carcass of these cultured aquatic animals, suggesting that this fermented sweet potato promotes better deposition of omega-3 fatty acids in the flesh of these aquatic animals. We also, the data also suggests that the pathogenic bacteria, total vibrios, were decreased in the gut of animals receiving the fermented sweet potato meal. But there is a significantly increased population of lactic acid or probiotic bacteria in the gut of these animals. We found out also that the fermented feed ingredient exhibited an antibacterial effect when tested in vitro condition. So the fermentation product may produce a low molecular weight compound that is inhibiting the growth of pathogenic bacteria in the gut of these farm aquatic animals. So another product, fermented biomass, that has a significant potential to replace soybean in fish meal in the diet of aquatic animal is fermented copra meal. This product is developed by biotechnology at UP Los Baños, and we tested this one and optimized the inclusion of this material in the feed of aquatic animals. So PECM basically is fermented copra meal, protein-enriched copra meal. So this improved the digestibility of copra meal and increase the protein content of copra meal to a point that it would equal the protein content of soybean meal. Soybean meal is basically imported. So the development of PECM would somehow lessen the importation of soybean meal and the utilization of soybean meal to be used as a feed component in aquatic animal feeds. So soybean meal nutritive value is basically equal to that of PECM. Fermentation has significantly improved the nutritional value and feed value of copra meal. to a point that it is already equal with that of soybean meal. So we formulated diet containing 20% of this PECM to shrimp, tilapia, and milkfish, and we tried this one in a actual pond production trial, comparing the performance of animal fed with PECM to those that are fed with the commercially available diet from the market. So these are the ponds by which the animals were stocked. And at the end of the trial, we found out that the production yield of the animals fed with PECM is equal to that of those receiving the commercial diet, to about 2.2 tons per hectare. The FCR is also similar, and all other biological indices, yield, survival, productivity, is similar, indicating that the use of PECM is now feasible and could be comparable with the commercial diet that utilizes fish meal and soybean meal as the main protein sources in the diet. But PECM is fermented product, it is cheaper, it is easy to produce, and you do not wait a longer period of time to produce PECM as compared to the production of soybean that it requires pesticides, a lot of water to farm soybeans. So fermentation basically is the future of sustainable feed for aquaculture. So in terms of milkfish, we did a similar test comparing commercial diet with that of the PECM formulated diet. And the yield basically in milkfish is higher as compared to those receiving the controlled diet, indicating that PECM promotes better milkfish growth as compared to the commercial diet. So the yield is about 2.98, almost 3 tons, as compared to the controlled diet that the yield is about 2.5 tons per hectare. The growth rate is higher, recovery is higher, is similar. The feed conversion is basically better in those receiving the PECM diet. Again, this tells us that PECM is better than commercial diet in the milkfish trial. So the next topic that we'll be discussing is the use of bioactive carbohydrate coming from natural sources or organic bioactive carbohydrates as feed supplement for aquatic animals used in aquaculture. So basically, disease resistance and fast growth is highly desired in aquaculture. Diseases are common in cultured animals because animals are being cultured in a high dense system by which stress is normally experienced by these animals. And also, this stress could result to inhibition of growth. So, strategies and feed additives that promotes better growth and lowering of disease risk in aquaculture is highly desired in farming of aquatic animals. So bioactive feed additives are natural organic compounds that are natural in source or in origin, and it could influence the physiological processes that could result to improvement of the overall biological performance of farm aquatic animals. So the first ingredient that or the first supplement is the bioactive. So this pocoidan is extracted from biela, brown seaweeds that are common to Australia. So this Dorbiela pocoidan is sourced or is produced from a seaweed that is common to Australia. And we tested the inclusion this pocoidan. We included this material or this supplement to shrimp diet, Pineus baname, ranging from 0.05 to 0.4% % of the diet. And we found out that dietary inclusion level of 0.2% of the diet significantly increased the growth rate of the animal to about 40%. So there's an enhancement of growth to about 40% in supplementation level of 0.2% of the diet. We also investigated the response of these animals that have been that are maintained with diet supplemented with fucoidan when they're exposed to major pathogens affecting shrimp aquaculture in the Philippines, the white spot syndrome virus and the Vibrio paralyticus infection. Vibrio paralyticus is the causative agent of early mortality syndrome. And we found out that a dose of 0.2% promoted 80% survival of Pineus baname when exposed to WSSB virus. Similarly, 90% survival could be attained in animals receiving the fucoidan when they are exposed to Vibrio paralyticus infection. So, this tells us that dietary supplementation with fucoidan could promote growth in Pineus baname. At the same time, it could increase the survival of these animals when infected with white spot syndrome virus and Vibrio paralyticus pathogens. So not only that growth is promoted, it could also increase the resistance of animals against these pathogens. So another polysaccharide that we have tested is an acidic polysaccharide that we have extracted from the green seaweed Ulva lactuca. So the animal that we tested here is Sila cirrata. This is the world fastest crustacean being cultured. This animal could grow from 5 grams to 750 grams with a period of 5 months culture. This is one of the fastest growing crustacean being cultured in the world, and this is found in the Philippines. Currently, the potential of this organism for aquaculture is highly significant. However, when cultured in the actual pond conditions, they are usually affected by white spot syndrome virus at the early phase of culture. And we investigated some compounds from seaweeds if we could enhance their resistance against white spot syndrome virus. So we developed a protocol to extract acidic polysaccharides from ulva and incorporate this one in the diet of juvenile Sila cirrata. And we tested inclusion levels ranging from 0.1 to 2,000 kilogram per 2,000 grams, 2,000 milligram or 2 grams per kilogram diet of the mud crab. And we found out that the optimum dose to promote or to enhance disease resistance and immunological responses of the crab in terms of propanol oxidase, respiratory burst, and antibacterial activity of the hemocyte is at a dose of 1,000 milligram per kilogram diet given every 3 days. If the dose will be higher than that, there's a significant decline in the capacity of the animals to resist the infection, or there is an immune suppressive effect if the compound would be given higher than 1,000 milligram per kilogram. So we tested this one incorporated this extract to the diet of juvenile mud crab. And after the feeding trial, we exposed the animals to white spot syndrome virus pathogen. And we found out that 80% enhancement in survival is observed in those receiving the ulva polysaccharide as immunostimulant as compared to the control after the 30 days of infection trial. 80% survived in the treated group. So, we also look at the enhancement of the digestive enzymes because we are delivering the compound via dietary means, and we found some growth promoting evidence of this compound to mud crab, and we look at what is the mechanism that could be related to growth promotion. And we found out that all digestive enzymes, including amylase, lipase, and proteases, were 30 to 40 times enhanced in the animals receiving the immunostimulant coming from ulba. And also, the cellular energy allocation, a measure of the total energy or the total nutrient deposited on the cells of the animal, is about 40% better in crabs receiving the immunostimulant, indicating that the rate of anabolism or tissue synthesis is promoted. So growth is basically promoted with the addition of this immunostimulant in mud crab juveniles. So another compound that we have tested is peptidoglycan. Peptidoglycan is a cell wall component of gram-positive bacteria. So the source of this one are gram-positive bacteria, and this could be produced by fermentation, and it is available in a sustainable manner. So peptidoglycan could be available and could be used in mass scale for aquaculture. We tested previously, we have optimized the dose, and we found out that it is 2 grams per 100 grams. That is optimum for Pineus monodon. And we added that one to the diet of Pineus monodon, controlled and treated, and we found out that in terms of weight gain and nutrient deposition, those that receive the peptidoglycan diet has better FCR, weight gain, and deposition of protein and lipid in the tissues. So it promotes better growth, better tissue deposition of protein and lipid. At the same time, we found out that the muscle fibers of those receiving the peptidoglycan are bigger as compared to the control group, indicating that the growth promotion is due to muscle hypertrophy or muscle growth in shrimp. So we look at also at the amylase or digestive enzymes. It is upregulated to about 30% in animals receiving the peptidoglycan diets. In terms of lipase also and digestibility, we found out that the overall nutrient digestion in those receiving the immunostimulant are significantly better, or it promotes better digestion of nutrients. And it may explain the better growth performance and better muscle growth in the immunostimulated animals or those that receive the peptidoglycan diets. We also exposed this shrimp to white spot syndrome virus infection to know whether animals that could grow faster or with exhibited better growth are also resistant to viral infection. And we found out that it is 100% resistant to white spot infection. The rest of the treatment were all dead after 10 days upon the exposure to white spot syndrome virus, but not the peptidoglycan treated group. It exhibited a survival similar to the negative control that are not exposed to the white spot syndrome virus. 100% survival had been attained by peptidoglycan supplementation. Another compound that has a growth promoting effect on shrimp is chitin. Chitin is a component of shells of crustaceans. So these polysaccharides are composed of glucosamine, and this comprises the shells of all crustaceans. So we tested if adding chitin to the diet of Pineus baname, whether it could lower the optimum protein requirement of this shrimp, since shell formation in crustacean requires protein to be catabolized and to be assembled as shell. So basically, the shell of the crabs are synthesized from carbohydrates and protein. So we hypothesize that if we add chitin or the precursor of chitin to the diet of these animals, this shrimp would no longer catabolize protein to be used as the component of their shells. So we tested different inclusion levels of chitin to Pineus baname at 0.5 and 1%. And we compared two diets with different protein levels. The optimum dietary requirement of Pineus baname is 32 to 35% protein in the diet to attain optimum growth. But we dropped that one to 27% and added chitin to the diet. We want to see if we could lower the protein requirement of Pineus baname without negative effect on the growth of these animals by adding chitin to the diet. Our results suggest that animals receiving the 32% protein without chitin exhibited similar growth to the shrimp given 0.5% chitin added to a 27% protein diet. So what is the meaning of this result? It tells us that we could lower the protein content in the diet of Pineus baname without sacrificing the growth performance. We just add 0.5% chitin to the diet. And the animals would have a maximum growth even though that the diet has a lower protein content. This is an efficient way to lower the protein content in the diet of Pineus baname because the cost of feeds in shrimp aquaculture is basically dependent on the amount of protein. Protein is the highly priced ingredient in aquaculture. The lower the protein we use in the diet, the cheaper would be the feed material. So this tells us that adding chitin could lower the protein incorporation or protein requirement of Pineus baname. And it may lower the cost of feeds for Pineus baname. The feed conversion is not affected. So we could lower to about 5% protein in Pineus baname without negative effect on the growth and feed conversion of this animal by only adding 0.5% chitin on the diet. And chitin is basically a waste material from the crab and shrimp processing industries. Shells are just thrown as waste byproduct of these industries. And it could be utilized, processed, in use as a feed ingredient or supplement for Pineus baname aquaculture. So another material that so far we have tested to promote better growth and efficient nutrient utilization in cultured animal is the bioactive algae-based feed additive from Ecolife G1. This is marketed by OB Trading, and this material is a seaweed-based processed bioactive algae. So we incorporated this material in the shrimp diet, Pineus baname, at a rate ranging from 0.5 until 10% of the diet. And we look at whether it could improve growth or what is the optimum inclusion dose to promote efficient and better growth of the shrimp. We found out that at a dose of 0.05% or at the dose of 0.5% in the diet, it promotes 80% growth improvement in Pineus baname. And all of the dietary doses, it promoted better growth. And there is no decline in growth even at the higher inclusion dose of 10% of the diet. But the lowest dose to promote 80% improvement is about 0.5% of the diet. So this figure shows us the obvious growth size difference between the control and the treated group. the 0.05% group is exhibited a bigger size, are bigger as compared to the control group. And we look at the effect of the treatment on the muscle transcription of muscle genes that are responsible for muscle growth. And we found out that alpha-actin in heavy myosin chain, the major component of muscle fibers, it is twofold enhanced. These genes are twofold enhanced as compared to the control group, meaning higher transcription of these genes are exhibited in the treated group as compared to the control group. And the target of rapamycin gene, another gene responsible in promotion of muscular growth in shrimp, it is also, the transcription is increased to about twofold also. So all of these are telling us that the better growth is associated with better muscular growth in this animal. So supplementation with Ecolite G1 may promote better growth due to the increase in muscular growth of the shrimp. We also look at the stress tolerance index of animals given with Ecolite G1, and we found out that it is 50% better as compared to the control group. So faster growth, better stress tolerance are positive factors that could be attained by using Ecolite G1 as a supplement to this animal. We look at the immune-related genes on the shrimp, crustine and pinidine. These are antimicrobial proteins. These are used by crustacean to eliminate microbial and bacterial infections. And it is twofold enhanced. In pinidine, it is about threefold enhancement. So not only that growth promotion are manifested by supplementing diet with Ecolite G1, but also the immune responses of the shrimp are activated, and the stress tolerance of these animals are also enhanced. So overall, this tells us the lesson that we could learn from this talk would be microbial biomass through fermentation could be a source of sustainable feed protein for aquaculture, and we could lower the utilization of fish meal and imported soybean meal by going into the mass production of fermented biomass to be used as alternative bioactive feed component for the Philippine aquaculture. And I am encouraging investors to invest on the production of microbial biomass, fermented biomass for aquaculture feeds. Secondly, bioactive polysaccharides of natural in origin, such as peptidoglycan, fucoidan, chitin, and ulvan polysaccharides could improve productivity, efficiency, and profitability of major aquaculture species in the Philippines. So utilization of this compound may result to improve the competitiveness and the enhancement of aquaculture production in the country. So hope that everyone has learned from this talk. Thank you very much. This is the end of this talk. Thank you.
Maraming salamat, Sir Rex, for that great presentation. Itong mga natural at organic feed additives na ito pala ay isolated mula sa seaweed at aquatic microbes.
Yes, and as we recall, ang purpose nito ay growth enhancers, metabolism boosters, and stress and disease resistance activators na maaaring idagdag sa mga aquatic feeds.
Tama. At para naman sa ating mga live audience, again, you can type in your questions to the comment section at sasagutin iyan ni Sir Rex later sa ating open forum. So type in your name, your affiliation or location, and then your question.
So at this time, we are now down to our third presentation. It will be presented by a professor at the Institute of Oceanography and Environmental Science and the research director of Mindanao State University, Tawi-Tawi, College of Technology and Oceanography.
She is an expert in marine ecology and received her PhD in biology at Florida International University. Her research interests include marine plants, seaweeds, and seagrasses. To talk about the Seaweed Research Center in BARMM, let us welcome Dr. Siti Zaida B. Saloon, live from Tawi-Tawi. Morning, Ma'am Zaida.
Good morning, ma'am.
I think, Ma'am Zaida, are you there po? I think we're just having a bit
Hi. Magandang umaga po sa lahat ng nanonood sa ating sa umagang ito. Ako from Tawi-Tawi.
Hello, ma'am. Ma'am Zaida, may tanong lang po kami. Ano-ano pong mga technologies and facilities ang nandito sa ating Seaweed Research Center?
Sa ngayon, meron po kaming 3 laboratories. Meron po kami seaweed tissue culture lab, kung nagpo-produce po kami ng mga bagong seed stocks para lumago ang ating seaweed industry, hindi lang dito sa farm, sa buong bansa din. Pasensya sa Tagalog po. Tapos meron din kami molecular laboratory para ma-character namin ang mga seaweeds na na-collecta from the wild tsaka sa mga farms. Okay. At meron kami post-harvest process lab na kung saan nag-extract na kami ng carrageenan at titignan namin ang quality ng carrageenan extract from different farm varieties tsaka wild din. Thank you.
Thank you, Ma'am Zaida. At dahil dyan, excited na po kaming makinig sa inyong presentation. Alam kong marami tayong matututunan. So take it away, Ma'am Zaida.
Thank you.
Hello everyone, today I'm going to talk about our work at the Seaweed Research Development Center in the Bangsamoro Autonomous Region in Muslim Mindanao. SEERDEC is funded by DOST under the Science for Change program. The farming of the red seaweed started in the 70s and is now a major source of livelihood for coastal communities in the country. The 2 most commonly cultivated species are Capophycus alvarezi and Eucuma denticulatum. Most farmers prefer Capophycus because it actually commands a higher price. Capophycus, also known as cotton eye in the industry, costs around 150 to 170 pesos per kilogram as raw dried seaweed, while Eucuma costs around 25 pesos per kilogram. Now, this is a farm in Panglimasugala, Tawi-Tawi. They use the off-bottom method. The seaweeds gets exposed during low tide. So in shallow areas, they use this kind of farming method. It's a pretty extensive farming area. And then in Sitangkay, they use the floating method. They have around more than 34,000 hectares of seed farms. So they use plastic water bottles as floaters, and they need a small boat to go around the farms. This is just to show you the small boat. This is a farmer who just harvested his seaweeds. So seaweed farming is a family enterprise. Usually for example here, the mother is being assisted by her children to tie the seedlings on the lines to be planted the next day. Unfortunately, apparently the seaweed farming community does not consider, they don't account for the inputs of the women and children into the seaweed farms. if you look at it, they contribute a lot in terms of labor costs because they actually help in the preparation of the seedlings and during the maintenance of the farm and harvesting and drying of the seaweeds. Now, the commercial farming of red seaweeds has increased over time because of the global demand for carrageenan. Carrageenan is a polysaccharide that serves as a thickening, gelling, and binding agent in the food pharmaceutical and nutraceutical industries. If you look at it, for example, your burger, cake, beer, milk, cheese, a lot of our food products actually today contains carrageenan. And of course, in your personal care products, lotion, toothpaste, cosmetics, and pet food. So it has a lot of applications in different industries. But although the number of farms have increased, the seed production of the country has declined since 2010, and Indonesia's production has increased. In fact, in 2019, Indonesia produced about 9.3 metric tons with a value of more than $1 billion, while the Philippines just contributed about 1.478 metric tons with a value of $207 million. So there is a need to address the problems facing the seaweed industry. Now, the decline in seaweed production seems to be partly caused by the seed stock selection procedures of farmers, wherein they propagate plants already in culture. And the repeated vegetative propagation of the same plants has led to a decrease in genetic diversity and problems associated with low-quality seed stocks. They become susceptible to ice disease, such as with extreme changes in temperature and salinity. They become susceptible to ice disease, and they start the cell lines turns to whitish in color and tries to disintegrate. Unfortunately, there's no treatment for it until now. Although I'll talk later about what the farmers claim to be possible treatment. And then you have epiphytes grow on them, and then this is a calcareous algae that grows on seaweeds and constricts their growth. And the ice disease and the low growth rate, all of these actually also lead to low carrying yield and quality. Now, there's widespread use of fertilizers. Apparently, farmers claim that the fertilizer makes their plants grow faster and prevents ice disease. And they say that if they stop using fertilizers, their plants will die and stop growing, and they will have ice disease. But they also complain that it costs a lot in terms of money because they have to buy the fertilizer, and it takes a lot of time to dip, because they call it charging, to dip their plants in the fertilizer solution and then take them out and leave them overnight covered by a tarp, and then plant them the next day. So they do this every 15 days. And no amount of convincing, arguments that we use against the use of fertilizer, they won't give it up because they said they tried it and it didn't work. The plants died. So we need to provide them an alternative, that is new strains that do not require the fertilizers and can still grow in the same rate as what they claim, because they can now harvest them in less than 30 days and then sell them. Another problem is the traditional drying method exposes actually the seaweeds to dirt. For example, they just hang them or put them on a platform or lay them here on the ground with just a tarp protecting the seaweeds from the ground, so they get exposed to dirt. And then processors and traders actually complain because it's additional cost on their part, because they have to pay people to clean the raw dried seaweeds before processing. If you look at the regional seaweed production, you'll see that the BARM contributes about 48%. The second, these are the four major producers of seaweeds. You have Mimaropa is the second major producer, followed by the Zamboanga Peninsula and the Visayas at 6%. Now, for the BARM, Tawi produces more than half of that, the 48%, and followed by Sulu at 30.88%. So we propose for the establishment of the Seaweed Research and Development Center to address some of the issues faced by the seaweed industry by optimizing the seaweed value chain and creating and promoting a commercial, sustainable, cultivated seaweed industry in the BARM by producing quality seed stocks, improving health and farm management, and producing high-quality raw dried seaweeds. There are 3 component projects. Project 1 is optimizing laboratory and land-based nursery culture technologies of aquaphytus and eucuma, led by Professor Karen Sirag. Project 2 is working on the molecular characterization, selection, and production of eucumatid cultivars. And Project 3 is optimizing post-harvest handling of aquaphytus and eucuma. So we collected wild and farm aquaphytus and eucuma from Tawi-Tawi and Sulu, and then we brought them to the lab for propagation using tissue culture techniques, and we're actually doing strain selection based on growth rate and survival rate. And the best performing are then propagated in the sea-based nurseries. We also do selection at the sea-based nurseries based on growth rate, survival rate, carrageenan yield and quality, and resistance to ice disease. At the end of the project, we aim to distribute. We want to distribute the best performing strains, okay, to improve and boost production of the seaweeds in the BARM. So we also analyzed carrageenan yield and quality of those samples that we collected, and we did molecular characterization and phenotypic analyses of the different specimens that we collected. Now, just to show you, these are some of the farms and wild areas where we collected samples. So we went to the Latanaro in Sibuto. This is Mandulan in Bunga, and this is in Siasi, Sulu. But also, this is a macroalgal bed, pretty extensive in South Lubi and Tawi-Tawi, but it's mostly it's dominated by Eucuma denticulatum. We found most of these seaweeds, eucumatoids, growing in transition area between a seagrass dominated and a coral dominated bed, and they're usually subjected to strong waves and strong current. They grow on coral rubble. Okay. Just to show you, this is the wild Capophycus. They have like thinner thalli, like at the end, the tips are actually narrower than those that are in the farm Capophycus varieties. And these are the wild eucuma. They're bushy-like. Okay. And this is the carpet Capophycus. They have mandibulate structures that contain the spores. So we collected all of this and brought them to the lab. So we now have a distribution map of wild Capophycus and eucuma denticulatum in the Sulu archipelago. So the red and green dots are areas we surveyed, but we only found them in areas with the red dots, indicated by the red dots. And the asterisks there actually are areas where we found the Sucarpid Capophycus. Unfortunately, we could not identify the eucuma denticulatum, Sucarpid eucuma denticulatum, so we were not able to collect any. One interesting finding was we found this wild Capophycus striatus in a mangrove stand in Siasi. They were growing in, it's a mixture of so it's dominated, the mangrove stand was dominated by Cineracia alba, and there were also seagrasses growing in the area. And we found the Capophycus growing on top of the seagrasses, and some of them were attached to the pneumatophores, and they were exposed during, low tide. Unfortunately, none of the samples we brought to the lab survived. Apparently, they would not grow in the conditions, the normal conditions where you find most sites where you find Capophycus. Perhaps they have adapted to the extreme temperature and salinity in the mangrove stand that they could not adapt to the other conditions where you would normally find Capophycus. We also, this is just to show you the morphological profiling work of the different samples that we collected. We now have a guide to the eucumatoids of the Sulu archipelago. The guide is being reviewed. We were able to collect 79 cultivars, but we grouped them into 52 groups. For example, here, this is Capophycus striatus, and it's cultivated in Tawi-Tawi. And they call it 7 colors because of the different colors. 7 colors, they call it the 7 colors in one plant. Okay. But Compicus and Eucheuma are morphologically plastic, so we can't tell if the 79 or the 52 groups that based on morphology and color are different from each other, or they're just morphological different, but actually they're the same, similar genetically. So we have to wait for the results of our genetic analyses. And for the wild, we couldn't identify them to the species level, so we just put graphite. We will have to wait for the genetic data. This is for Eucheuma lenticulatum. They call it China. Okay, I don't know why they call it China. And they have funny names actually for the different varieties. They have Marimar. They call it, one was they called it Bitsy Bitsy because apparently the person who actually first cultivated the variety was able to buy a Mitsubishi motor for his boat. So yeah, they call it Bitsy Bitsy. And we have larger specimens were deposited at the MSU Herbarium, and another set will be deposited at the National History Museum as required by BFAR when we asked for the gratuitous permit. So we were able to establish a molecular genetics laboratory, and we are now analyzing, I guess, genetic makeup of our seaweed collection. We also expanded our seaweed culture laboratory, so we now have around 11,000 cultures in the lab. So it takes a lot of hands to actually maintain the laboratory. So we do a branch micropropagation and growing the spores that are released from the sister carpet Capophycus, from the mammillate structures here. And we have been documenting sporting development in the lab for Capophycus. So from 12 hours to 24 hours, and then the rhizome formation, apical subapical development, and then of course until they reach around 15 months old, and then they're ready for outplanting in the hatchery and sea-based nursery. So these are the current cultures in the laboratory, around 11,000 cultures of farmed and wild seaweeds, Capophycus and Eucheuma. So the land-based nursery, we were able to propagate around 1,993. These are laboratory-generated seedlings using branch micropropagation and sporelings. We were only able to transfer 1,984 laboratory-generated seedlings in the nursery because not all of those from the hatchery survived. So this consists of about 30 farm varieties and 42 wild Capophycus species are planted in Cebu. So this is Capophycus species seedlings generated in the lab of spores collected from Taduba, Sawi-Tawi. The average specific growth rate is actually comparable, about 6.67% per day, comparable to those reported by Hurtado et al. And Mustafa. This is so we were able to establish a seaweed post-harvest processing laboratory where we now assure moisture content of the raw-dried seaweeds, and of course we quantify the foreign matter in the raw-dried seaweeds collected from the farmers and traders. The farmers are actually requesting us to analyze their samples for moisture content so they could bargain with traders on the price of their RDS. We also extract native and alkali treated carrageenan using a modified carrageenan extractor. So this is actually the carrageenan, fresh carrageenan we produce, and this is the dried carrageenan that we have in the lab. We also analyze viscosity, the texture of the gel, the gelling melting point, and CRSs of the carrageenan extracted from the different samples. We have been pilot testing the UPLB Biomech Solar Assisted Seaweed Dryer, and we initially showed a 35% decrease in drying time, which is very important for Tawi-Tawi because it rains from like 12. It's raining from 12 to like 20 days in a month, so that's a lot of rain. So farmers usually just cover their seaweeds, but then It takes a long time to actually dry their seaweed, so it compromises the quality, and processors then complain about the quality. So a solar assisted dryer would actually help them a lot. So if you look at the quality of the raw dried seeds of from farmers and traders, you see the moisture content range from 38.92 to 39%, which is about higher than the 32 to 35% set by BIFAR standard. Okay. And then you have the foreign matter content is from 7.32 to 8.11. Most of these are dyes and like gravel or sand, okay, that gets mixed up with the raw dried seaweeds, which is higher than 1 to 3% standard set by BIFAR. In terms of average carrageenan yield and quality of Caulophacus alvarez and tawi-tawi, the look at the carrageenan yield is comparable to what was reported by Simatupang et al. In 2021 for Indonesia. These are different islands. Actually, I think around 11 islands in Indonesia. And then, but if you look at the viscosity, it's a lot lower than the report 35 to 183. And they didn't report the syneresis, but the gel strength, you look at it, it's like 291 to 735, but while the gel strength of the carrageenan from tawi-tawi of the Caulophacus from tawi-tawi was only 52.87 to plus minus 51.24. So, sorry, that's actually 5.24. So if you look at this, it seems that the quality of raw dried seeds from tawi-tawi is a lot poorer compared to other places. So there is a need to improve post-harvest processing of seeds from tawi-tawi. Now, we also do consultation meetings with farmers and document their harvesting, drying, and storage practices. We want to ensure we know the problem before we even help our LGUs to formulate policies to address these issues. So just like to point out, this is actually farmers in Tapaan, where seaweed farming started in the country. And this is Tapaan Island, the shore across Tapaan Island. And it's a beautiful island. It's just it's kind of hard to get there because there are no regular transportation to the area. So we have been going around this tawi-tawi, okay, and interviewing our farmers. We also do consultation meetings with LGUs and other stakeholders, okay, because we need their support, okay, especially for pushing for policies that will improve the seaweed industry in the barn. We have been conducting trainings and OJTs for on-the-job trainings for our students, and we also get people actually visiting, not just locals. We have even foreigners going there to visit our labs. We now have regular trips, like study tours for students of their laboratories. And then we, of course, have collaborations. For example, we have JICA, it's a JICA-funded project. We have AMOA with the Ateneo de Davao Marine Renewable Energy Center. We also provide technical support to EU-funded projects, and we are part of the Provincial Seaweed Council. And we just created the Provincial Management Office for the seaweed industry in Tawi-Tawi. So future direction, we see that we'll have a database of agronomic traits and environmental requirements of different Caulophacus and Euchima denticulata strains. We hope to package aseptic seed stocks in partnership with the DA, BIFAR, and MAFAR. I had a discussion with Dr. Fred Santianes about this. It's important that when we distribute in the future seed stocks produced in our laboratory, it won't go through the farm. We just have to go from lab and then distribution to the farmers in other areas of the country to make sure we don't distribute pathogens that from Tawi-Tawi and then spread this around the country. Or if we may have it if we start producing also in other labs in the country, okay, if we have the technology to make sure that we distribute aseptic seed stocks. And then have development of culture technology for other economically important seaweeds, like ova, um, gracilaria. Um, and we also have to diversify the derived products from eukaryotes because there are so many applications. There's huge demand for it. So I think we need to not just focus on carrageenan, but other bioactive compounds. We also need to start producing, especially tawi-tawi, seaweed-based food, personal care products, and cosmetics. One of the things that the provincial government in Tawi-Tawi wants to do is we want to have like a same like every time you think of Tawi-Tawi, you think about a seaweed-based product that Tawi-Tawi will be known for, like around the world. Hopefully, we will have that, as we say in Tawi-Tawi, inshallah. And so please like our page, our Seaweed Research and Development Center Facebook page. And on behalf of the research team, I'd like to thank everyone for listening to me today. Thank you.
Thank you, Ma'am Zaida, for sharing with us the researches and developed technologies, as well as the goal of SEERDEC, which is to increase the seaweed production in the Bangsamoro Autonomous Region of Muslim Mindanao.
Malaking tulong po talaga sa ating mga seaweed farmers itong mga bagong strains na ibibigay sa kanila. It will improve their production and will promote sustainable seaweed farming. Bukod sa resistance nila to certain infestations and diseases, makaka-adapt pa ito sa ating nagbabagong klima.
And with that, we hope na handa na ang ating audiences sa kanilang mga katanungan dahil dadako na po tayo sa ating open forum kung saan live nasasagutin ng ating mga experts ang inyong mga tanong at komento tungkol sa mga ibinahagi nila sa atin ngayong umaga.
To start our open forum, tawagin po nating muli sina Ma'am May, Sir Rex, at Ma'am Zaida. We also have here with us Ms. Fidelia Flor C. Mero, our Senior Science Research Specialist from the Inland Aquatic Resource Research Division, to be our moderator. Hi, Ms. Ella, and welcome back po sa ating mga experts.
Hi po.
Hello po. Good morning. Thank you, Dana and Danica. So good morning po, Ma'am May, Sir Rex, and Ma'am Zaida. Handa na po ba tayong sagutin ang mga questions ng ating live audience?
Yes po.
Yes.
I think we're ready.
Yes, kung handa na po kayo, let's
Good
begin.
morning.
Yes, let's begin po our first question. So this is for Miss Mayurat, Ma'am May. May nag-comment po na napakaganda po ng presentation and I hope maging available ang mga varieties sa PhilRice Station para may option na po ang mga farmers for a healthier rice varieties. So galing po ito kay Elmer Caballes. So thank you very much for your comment po.
Yes, sir. Yun din po yung aming vision na ito ay maging available. Hindi lang po sa lahat ng PhilRice stations but all throughout the country. So far, nasa stage pa lang po kami ng pag-validate po ng ating mga lines. So magiging available ito hopefully in 8 years time. Yan po.
Okay, thank you, Ma'am May. So for our next question po, this is for Dr. Rex. What is the price po of the products, the formulated products? Meron po bang mga side effects na nakita nyo or observe? And can the products be used for livestock? This is from Chester S. Libong from La Union. Dr. Rex?
I think these products could be applied or used in livestock because, for example, the EcoLife G1 previously it was marketed and developed for livestock, basically sa manok po at saka sa nangingitlog nating mga animals. Now only recently that we tested this one for aquatic animals, and in terms po of pricing, it may be in the range of 50 cents until 1 peso per kilogram of feed because napakaliit lang po ng ating madagdag and it is highly significant po yung effect nito sa ating mga inaalagaang aquatic animals. But then again, this is also applicable for livestock because previously it has been optimized also and applied for livestock. For Ecolab G1, hanapin nyo lang po yung OB trading, and for the Fucoidan because ang previous studies nito na-try na to sa race horses. Ginagamit to sa mga racing horses. And we found out na napakalaking impluensya nito sa pag-protect ng ating mga sugpo sa sakit na dulot ng white spot syndrome virus, the number one dreaded viral disease that is being a problem in the Philippines. So, ang cost nito range from 50 cents to 1 peso po per kilogram ng feed. Yan lang madagdag but significantly malower ang risk ng disease occurrence. At the same time, mapatagdag income po dahil mamagnify ang income, tsaka ang production, tsaka productivity ng ating inalagaang mga aquaculture animals po. Yun lang. Thank you.
Thank you, Dr. Rex. So I hope that answers your question, Chester. So for our next question from our live audience, this is for Dr. Zaida. Paano po tumutubo ang seaweed at paano po ito napapanatili ng maayos ang paglago nito? This question is from Anthony Malyari of Balintocatak Integrated School, Grade 10 Mercury. Dr. Halun.
Good morning, Anthony. So, sa wild, lalahin na muna natin sa wild, ang seaweeds ay tumutubo sa mga coral rubble, sa sediment, tapos malapit siya, actually in between siya, nakikita siya kung pabili ka sa seagrass, saka coral, nasa transition, sa gitna siya ng coral dominated, saka seagrass dominated, at malakas yung agos, doon na gustong tumubo sa kung saan malakas ang agos. Tapos naman, sa farm naman, tinatali siya sa lubid, or yung nylon, tinatali siya doon at pinapatubo, pinahahang siya, tumutubo, mabilis tumubo doon, nakahang lang yan talaga, yung mga floaters lang. Kung nakita mo yung pinakita kong picture, imagine mo na isang line, tapos nakatali na isang tie-tie, plastic straw, para doon siya pinapalaki ang seedling. Ganun lang kasimple talaga, pagpapatubo ng seaweeds. Paano natin mapapanatili ang ayos na pagtubo ay yung sabi namin, gusto kami bagong seed stocks dahil yung mga seed stocks na ginagamit yun, kinakailangan gamitan ng fertilizer. Yung aming na-produce, very promising siya sa field ngayon, pero hindi siya pwedeng gamitan ng fertilizer. Kasi meron kami nilagay na nursery malapit sa may mga nagpo-fertilize, namatay sila. So, pero ililipat namin sa ibang lugar na walang nagpa-fertilizer at malakas ang agos, maganda ang tubo at 97 to 100% ang survival rate. So, merong promising ang mga seed stocks kung bigyan na natin sila ng bagong seed stocks na pwedeng gamitin na mababago natin yung sistema at paggamit ng fertilizer. Salamat.
Maraming salamat po, Dr. Zaida. At ang ating susunod na katanungan naman po ay para kay Ma'am May. Would eating low glycemic index rice help in lowering the risk of diabetes among diabetes patients? So, Ma'am May?
Yes, it would. But of course, hindi lang naman kasi yung kinakain natin ang cause sa atin ng diabetes or nagbibigay sa atin ng risk of diabetes. So we must always remember that diabetes will not only rely on what we eat, but a big factor din yan yung ating lifestyle. So, yes, eating low glycemic index rice or any other food na may low glycemic index would help.
Ayan. Thank you, Ma'am May. So our next question po ay addressed to Dr. Rex. Why there is a need to look for fish meal alternatives for aquaculture?
Kasi kailangan talaga because basically yung ating aquaculture is dependent fish meal yung feeds. Ang problema ngayon, we have declining catch even those fish that are used as food fish. So parang ang mga asosasyon ng mga feed millers, mga scientists, tsaka mga environmentalists sa buong mundo nagkaroon ng common agreement na dapat yung isda sa dagat ay para sa tao, para pagkain. Hindi na huhulihin natin ang isda sa dagat tapos ipakain din natin sa hayop. So it is an inefficient way to use natural resources. O harvestin mo tapos gawin mo ring pakain sa isang animal. It is an efficient way to utilize the resources. So kailangan natin talaga to utilize plant-based material to convert that one into aquatic animals as feed. So kailangan talaga na ang ating isda sa dagat man o sa lawa o sa ilog, pagkain ng tao yan. Hindi natin dapat gawin yan as feed para sa isda or para sa livestock. So we need to have a technology to convert plant-based biomass into a material na para magawang ingredient ng feed ng livestock man yan o ating mga isda. Hope na sagot po yung tanong, Miss Ella. Thank you.
Thank you, Dr. Rex. So we're now down to our last two questions po. So itong second to the last question is for Dr. Zaida. So what are the challenges in convincing the farmers to stop the use of fertilizers?
Ang I guess yung laging iniisipin natin kasi income yan, diba? Kabuhayan ng ating farmers. So ngayon, ang mga strains na ginagamit ng ating farmers ay sanay na mga 6 years, 7 years na yata sila gumagamit. Parang may mga sources on 12 pa na nag-start na sila gumamit ng fertilizer. So, hindi na tumutubo ang mga seeds nila kung walang fertilizers. So, paano makonvince sila? Kailangan natin ng alternative. Kasi hindi pwedeng sabihan mo lang na mag-stop kayo gumamit ng fertilizer. Wala tayong ma-provide ng bagong seedlings na pwedeng gamitin na malaki rin ang growth rate niya, mataas din ang growth rate at hindi naka-ice na hindi kailangan ng fertilizer. So, yung aming laboratory-generated seedlings, yun ang aming i-distribute sa aming farmers para change their minds pero kailangan may alternative. Yun.
Thank you, Dr. Zaida. Yan, may humabol pa po ng mga tanong. So, ang next natin question is for Ma'am May. Are there any health risks from eating irradiated rice?
Okay. Yung irradiation kasi is a way to induce mutation sa ating, not just in rice, but also other crops. Okay, and mutation is a natural process. So lahat ng organisms undergo mutation. Pag mga microorganisms, mabilis lang ang mutation rate. Pero pag mga higher organisms, mas babagal ang mutation rate. So we induce mutation in rice specifically para mas maging diverse yung population natin. And we want diverse population for breeding purposes. Pero yung irradiation na yun, effect niya lang yun doon sa rice. So when we eat this irradiated rice, wala siyang effect sa atin. So it's like normal rice lang din na hindi irradiated. So wala po siyang health risk.
Okay. Thank you very much po, Mami. So we are down po, may humabol ng question. Last question. So this is from Tagle Simplina. Currently, hindi po masarap ang lasa ng milkfish, tilapia at pati na rin ng cultured hipon, maybe because of the commercial feeds. Dito po ba sa organic feeds presented by Dr. Trifalgar, may improvement po ba sa taste? Malaking bagay po kung masarap yung lasa, therefore mas maraming bibili ng products. Dr. Rex?
Magandang umaga po. I think yung tanong ng lasa is perceptive. Hindi natin ma-quantify yan kasi it is iba-ibang tao, iba-ibang response. But posible din yan, how long was the product was harvested, gaano siya kahaba from harvest to the market, nag-apekto din yan. And for feeding perspective, it may appear it is still a subject for more research kasi ngayon yung mga research is mostly maximize lang po yung pagpalaki, pagpa-improve ng growth. Medyo wala pa po tayo anong epekto ng mga additives.
Na-mute po kayo, Dr. Rex. Dr. Rex, paki-unmute po.
Sorry.
Unmute ka.
Again.
Yes. Na-mute po kayo ulit. Dr. Rex, na-mute po kayo ulit.
Parang automatic nagmi-mute.
Ayan, okay na po. Doc.
Magandang tanong po to.
Mm-mm.
Ang lasa po kasi, relative po yan. Iba-ibang tao, iba-ibang panglasa. So hindi po natin alam kung nasa tao yung perception ng flavor na hindi malasa or kulang na yan ng MSG or something. Or pwede din po na processing na yan. I mean, how long was the product was available to the market since it was harvested? Isa din yan factor na umiibang lasa. Ang isa pa po is that wala pa tayo sa research area kung ano ba ang epekto ng feeding tsaka ng mga additives na to sa lasa ng ating alagang hayop. Hindi pa po tayo sa ganyang sistema ng research. We are not yet on that area. Sa ngayon po, ang ating in-optimize po yung production yield na muna. Paano padamihin, paano ma-maximize yung growth para ma-increase yung profit. But yung sa lasa po, medyo wala pa po tayo and wala ako masabing data on that kasi para wala talagang ginagawa na research on flavor ng aquatic animals. But that is a direction that we are looking in the future. Medyo inuumpisahan pa lang namin po yung ano bang epekto ng feeds sa flavor ng aquatic animals tsaka sa acceptability sa market. Salamat sa tanong, that is an area talaga na inuumpisahan pa lang po. Pero sa ngayon, wala akong masagot regarding that.
Thank you very much, Dr. Rex, at aabangan namin yan. So ito, may pahabol po na comment from our live audience. Sana po makapasyal kayo sa different coastal provinces like sa Ilocos region po. Marami po silang types of seaweeds that you might find interesting and study-worthy. So this comment is for Dr. Zaida.
Salamat. Actually, napakarami nga. I mean, hindi lang sa Ilocos region. Sana makapunta kami dyan. Kaibigan ko yung President ng MMSU. Sana makabisita dyan sa Ilocos. Pero nga, sa ngayon po ay dito muna po kami sa Barm area. Pero in the future po gusto namin po makapasya sa ibang parte ng Pilipinas. Thank you. And hopefully we will be able to see more of you. Thank you.
Thank you, Dr. Zaida. So that concludes our open forum. Thank you very much to our speakers and to our live audience sa inyong mga tanong. So now I would like to turn over you to our hosts.
Thank you as well, Miss Ella, for being the moderator of our open forum. Ang dami po naming natutunan. And of course, kung meron pa po kayong mga katanungan or gustong i-clarify sa ating mga experts, you can email us at picard@dost.gov.ph.
Yes. So finally, we are on to the last part of our NSTW Technofora. To show our appreciation to our experts, the Philippine Council for Agriculture, Aquatic, and Natural Resources Research and Development of the Department of Science and Technology would like to present the certificates of appreciation to Ms. May Rose Mawirat Abad, Dr. Rex Ferdinand M. Trifalgar, and Dr. Siti Zaida B. Halun for serving as resource person for the NSTW Technofora 2022 with the theme Ensuring Food Security, AANR Technologies, Busog-Lusog na Nasyon sa Agri-Aqua Innovation, held on November 24, 2022 from 9:20 a.m. to 12 noon via Facebook Live, signed by the Executive Director of DOST Picard, Dr. Reynaldo V. Iborra.
Again, thank you so much, Ma'am May, Sir Rex, and Ma'am Zaida for being with us this morning. Can we call again our speakers? Ayan, welcome back po, ma'am. Before we leave our audiences po, may gusto po ba kayong sabihin sa ating mga manonood ngayon, lalo na po sa ating mga magsasaka at mangingisda? First with Ma'am May.
Ayan, sa lahat po ng mga listeners po natin ngayon, especially sa ating mga magsasaka, Sana po ang aming product na low GI rice ay makatulong po sa inyo pag na-release na po yung ating variety dahil marami na po ang nag-aabang sa ating low GI rice na varieties. Ang una-una po makikinabang dyan ay ating mga magsasaka.
Yes, thank you for that, Ma'am May. And then, Sir Rex, how about you po?
Good morning again. Salamat. Gusto magpasalamat sa PICAART, sa DOST, sa oportunidad na ito na ma-share ko yung findings sa ating mga kasamahan dito at sa mga manunood natin. And gusto ko lang i-assure sa ating mga aquaculturists, sa ating mga mangingisda na hindi nagpapabaya ang ating gobyerno para mapa-increase ang produksyon natin para hindi tayo magkulang ng isda at pagkain sa ating hapagkainan. So again, salamat sa lahat, sa DOST at sa lahat ng mga nanonood sa event na ito. Thank you.
Thank you, Sir Rex. And lastly, Ma'am Zaida. Andiyan pa po ba si Ma'am? Ma'am Zaida?
I'd like to thank DOST PICAART for the invitation to share our work at the Seaweed Research Development Center sa MSU Tawi-Tawi. At gusto lang namin na makatulong talaga ang aming teknolohiya at saka yung aming mga seed stocks na magaling sa laboratory para sa mapalago ang seaweed industry hindi lang sa Tawi-Tawi, sa Sudu, kundi sa buong bansa. Maraming salamat po.
Thank you. Thank you, Ma'am Zaida.
Thank you po sa ating mga experts. At para po sa ating mga live viewers, we would like to remind you na maaari po kayong makakuha ng e-certificates sa pamamagitan po ng pagsagot sa aming CSF na matatagpuan sa link later na ilalagay sa screen at sa comment section. And make sure po na nakaregister po kayo. And there we have it. Now, to formally close the program, here to give us a message is the officer in charge of DOST PICAART's Office of the Executive Director for Research and Development, Dr. Juanito T. Batalon.
Distinguished guests, esteemed speakers and participants, ladies and gentlemen, good morning. In the celebration of the National Science and Technology Week with the theme Ensuring Food Security, AANR Technologies, Busog-Lusog na Nasyon sa Agri-Aqua Innovation, the Philippine Council for Agriculture, Aquatic, and Natural Resources, Research and Development of the Department of Science and Technology is deeply honored to have conducted this technology forum. I am very happy to see participants from different state universities and colleges, from the local government units and other stakeholders and practitioners who participated, listened to the presentations, and interacted by sharing the knowledge and experiences on the field of agriculture, aquaculture, and natural resources. With the three topics presented in this technology forum, I hope that the participants have gained knowledge on the advancement in agriculture, aquaculture, and natural resources research and development that would hopefully contribute to achieving agricultural and aquaculture productivity and provide livelihood opportunities and ensure food security. Your presence is an indication of your interest in science, technology, and innovation, especially in the AANR sector. I hope you find the presentations interesting and valuable in your respective endeavors. These topics were critically selected as these are among the interesting R&D initiatives that DOST and PICAART have supported. With that, I would like to thank our resource speakers for their unselfish commitment and dedication in sharing their bright ideas and knowledge, and to all the participants who took time out from their work and other equally important activities just to be here at the Technology Forum. I would also like to thank the organizers, the Inland Aquatic Resources Research Division, Marine Resources Research Division, Crabs Research Division, and Applied Communications Division of the DOST Picard, who have been working hard to make this technology forum possible. Again, thank you to everyone for making this technology forum successful and a memorable event. Mabuhay tayong lahat.
Thank you, Dr. Batalon, for your closing message. Nais din po naming magpasalamat sa lahat ng ating mga live viewers na sumamang matuto sa umagang ito. Sana ay maibahagi rin ninyo ang makabagong impormasyon na inilahad sa atin ng ating mga eksperto sa inyong mga komunidad.
Maaari kayong makakuha ng e-certificate sa pamamagitan ng pagsagot sa aming CSF or customer satisfaction feedback na matatagpuan sa link na nakalagay sa screen at sa ating comment section. Piliin ang Ensuring Food Security, AANR Technologies, Busog-Lusog na Nasyon sa Agri-Aqua Innovation sa ilalim ng activity title, sagutan ng form at ipapadala namin sa inyo ang inyong e-certificate sa loob ng 1 linggo. Magkita-kita po tayong muli sa susunod pang mga virtual events ng DOST Picard.
Muli, ako po si Dani Casembrano.
At ako naman si Dana Lee Logronio mula sa DOST Picard. Maraming salamat at Happy National Science and Technology Week.
Ating tandaan na ang siyensya ay hindi lamang para sa ating mga siyentista. Ito ay para rin sa ating mga mangingisda at magsasaka.